So this is going to be a scatterbrain talk. First of all, you have live plankton right here on the screen. This is coming from a stream in the device called Planktoscope. I can pass this around. This happens to be a culture from Tomales Bay. And you see just all kinds of stuff happen out in the ocean that we'll spend a lot of time about and talk a little bit about also just tools per se. And to really tell you this is live, I just turned the pump on, so who knows what you'll actually see. We could just watch the stream and just try to figure out, going opposite of what NOAA did, which is like, let's look at data. I'm going to talk about a lot of data, but I know you want to hear some synthesis. I watch this as TV at home, and I'm working on my kids a little bit. I think there are days that I can have them watch that stream for half an hour. I think somebody mentioned Foldscope. I just want to mention a few threads. You know, many people have known this project. It's scaled. I think I want to talk about scale. And in some sense, many of you don't know the stories. I want to tell 3 or 4 stories. The reason we scaled this project is actually because I got irritated by this tweet. We posted a paper on arXiv, and a somewhat known person tweeted about it. You can read that name. I'm not going to announce it. And what irritated me about it, they said, is a dollar origami microscope question mark. And I was stuck at the question mark. Did they not believe us? We had written a paper. As academics, we know we produce results, and we've shown all the data. And then it occurred to me, it's like, why should the world believe us? As academics, our goal is to bring change into the world. And that led to me putting on my website a little blurb that said, we're going to ship 100,000 microscopes to anybody in the world who wants it. We got 100,000 inquiries. I spent 300,000 of my startup money on shipping cost. I didn't tell my chair, and I didn't know what I was doing, but it mattered to me that it was not the tools, it was the communities that we were trying to build. And I think this is an important lesson, at least I've learned, in the context of democratization. At this point, it's the world's largest microscopy community in the world. Between 3 to 6 million kids around the world every year use these sets of tools, and it's growing. We're trying to build, all for a terrestrial context, a planetary microbiome, all done by citizens around the world. Some of you might not know we've been manufacturing now diagnostics to take the ideas that we learned in optics and apply to molecular biology. I do not understand why when cells and life is self-replicating, why do molecules cost so much? Lots of threads. This is a picture from Liberia where we've been looking at Lassa and Ebola. And one of the key things around this has been completely autonomous manufacturing. For every biologic, every enzyme that goes in, there is a trailer on campus that I've been eyeing for a while. So if you own that trailer, tell me about it. We've been designing an entire factory that's going to fit inside this that'll have E. coli production, protein purification, assembly of and we're trying to do this as a demonstration. I think this idea that capacity to manufacture products that are needed around the world needs to exist in some silos is absolutely broken. All of this is built on open IP, and so I can talk quite a lot about open IP at some point if people have time. And similarly, we've been trying to do this now, not just in the context of what happens in the field in hands of citizen scientists, but also scientists. The people you see in this photograph are Liberian scientists who've been working on an Ebola immunophenotyping project. Lots of people in Liberia never got a drug, but they still survive. We don't know the magical thing, what was in their immune system. And for the first time, by starting to put these instruments, and high-end instruments, now the little part of the AI part here is that the 2 instruments that you're seeing are completely autonomous AI labs that essentially we can program, and it starts to increase the throughput and capacity of these sets of things. So you should think of this as the humanoid embodiment, but for life sciences. Very general purpose instruments, we can install them anywhere in the world, and we've been starting to think quite a lot about how to increase research capacity around the world and not just in a few pharma companies. So I don't think you came here for all of this. This was just a blurb. I think you came here to hear about the ocean. One of the things that I want to mention to start with is how many of you have been to sea? I just want to get a raise of hands. So I think most of you I feel enormously small every time I'm at sea. And it just, all of this data, the fact that you can look around and you're just a tiny little thing, and out of that I'm going to take much amount of water I can take, it's only still a small portion. And I think it just kind of dawns on you how underrepresented much of the measurements are. Oceans are important from a context of carbon sequestration. This number gets tossed around between, 20 to 30 to 40% of all carbon that we emit is being absorbed by the ocean. And the process that we've been trying to understand is how does that occur? And it goes back to actually data and why it's important for us to scale measurements. That's the story I want to tell you today. The story begins with a crackling sound that came from a phone receiver. I don't know if you recognize this person. I am not this brave, but you have to understand that they were people that came before us. This is William Beebe right there, and Otis. They are hanging on this giant metallic sphere. This was the first person who crossed the 1,000-meter barrier. There is nothing else in this little giant metallic sphere other than one telephone wire and a cable. And then Gloria, on the other hand, is on the ship deck, and she hears in this crackling sound the very first time they lowered this, the term that Beebe used was marine snow. He said, "Oh, it's snowing down here." And Gloria, for a second, said, "Wait a second. What are they doing? Is this something psychologically as a joke?" But no, he meant it's snowing down there. And then, of course, much of what he wrote about was deep-sea fish. But really, that small little reference led to enormous amount of work for the next 50 years that we figured out what is called a biological carbon pump, which I'll talk about. If you think of it this way, is up on the top, you can see the atmosphere, essentially, we're starting to pump. And on the bottom is essentially the ocean sink that's taking in. As we put more carbon, the ocean sequesters more carbon. And I think this trade-off of how it does that is, in my mind, still open. And I'll tell you one nugget of the mystery for why it's actually open. If somebody shut down this biological carbon pump, we would have an instantaneous rise of essentially the carbon in the atmosphere. It's critically important. It's one of the scalable gigaton technologies that works on our planet, and I think we don't understand how it works. Much of the cell biology of this is driven by mountains of Doerr are one of the greatest examples. You can see this entire mountain is actually made out of 5-micron cells. These are coccolithophores, and I'm going to show you that in a second. They're phenomenal sequestration machine because they use biomineralization, so they make rocks and sink to the bottom of the ocean. And effectively, the simplest way of thinking about this is life in the upper ocean, which is euphotic. Essentially, as they die, they accumulate. We study viruses in this. Leads to these things called particles. These particles, because of gravity, would sink. And so the alkalinity pump in the ocean is 30%, biological pump is 70%. And people had assumed for the longest time, okay, we understand a lot of physics around sedimentation. We're going to just watch these particles sink. We can take photographs, and you could estimate the flux in the ocean. Here's the problem. This matter is living. So if I was to ask you that you drop a penny in the ocean, you can apply the Stokes' law and figure out how long it will take to fall into 4 kilometers. Fun homework exercise. It'll break your intuition if you do that. But if I drop a diatom, which is a living object, actually most cells have cytoplasmic densities 5 to 10% heavier than seawater, how long it would take. And until we did this work, and much of the last 5-6 years, this answer had remained unknown. So when we apply these types of physical principles, unknowingly we make these large mistakes. And I'm going to show you a little bit of data that it's not so simple. In fact, a diatom has a capacity in 100 milliseconds to change its buoyancy to essentially remain stationary, and it will not sink if it doesn't want to. The other part of this, and we look at the types of data sets that have been collected, there have been indirect measurements of this carbon flux. So when I pick up this is beautiful work, this is Cale's work. He measured all of the data sets that had been collected on size of these particles and their sinking velocity. Everybody who's done the Stokes' law knows, and then I look at this data, I could have fit any line anywhere in this data, frankly. That's the reality of it, and it just boils down to, again, it's not that somebody's not made the sets of measurements, but we haven't had direct measurements of sinking, and that's sort of kind of got me aggravated. It's like, no, I think it's time. It's so important. Why don't we just go out and start measuring this stuff? That was a couple years ago. One of the other aspects of this is, I'm not talking about this, but incredible amount of interactions. When I go out to sea at night, no filtration, this is what we are trying to understand. This is why ocean is hard. This is why it's important. You can think of hundreds of thousands of species interacting with each other, incredible food webs, motility. All of this biomass is rising and falling kilometers every day. The budget, the energy budget of this going up and down per day would actually be the US budget for an entire year. So an enormous amount of biomass travels. This is really some of the diversity that's out there, and I think since I'm running out of time, one thing I want to mention is in the kind of a climate discussions, microbiology has taken a backseat. And it's important to have models that make predictions, but it's also important to understand where are the nonlinear knobs. Biology is incredibly nonlinear, and one of the things that's starting to happen is its distribution is starting to change as a function of climate change itself. Okay, so I want to give you one nugget of a result. This is the classic Martin's curve. This is how we measure flux in the ocean. This determines how much biomass is going down. And one of the things that we had a sense for is unless you can measure the flux directly, it's very hard to actually predict what this curve is. But it's a very important curve in terms of making predictions of how sequestration work. We started 6 years ago just deciding we're going to spend a lot of time at sea. I missed a lot of faculty meetings and apologize for that. But it was very important for us to be at sea. And literally, we've done 17 expeditions so far in the last 6 years. All across the world. I think the only ocean that I've not been to is the Indian Ocean, which is ironic. I grew up in India. I'm working on that. If you've never been to sea, this is what it looks like. Sometimes these guys show up. And we decided to make cellular scale measurements at sea, which again is a problem if your entire world is actually shaking around. We solved that problem. And let me just show you a few sets of videos and data sets and discoveries we have made. First of all, when you go out to sea, you don't decide what you're going to discover. You watch. In the Arctic, with Kevin's lab and Ching as a postdoc, we actually have just discovered the lowest motility that has ever been reported in a eukaryotic cell. Minus 20 degrees Celsius, this is a completely new class of actomycin. Some of you know that our planet was completely covered with snowball Earth, and it implies that much of life had actually adapted, so just incredibly cold conditions, which we might not even see on this planet. Going back to the history of what I'm about to tell you, I've been working on schistosomiasis for a long while, so we often go out to Madagascar, we put waders on, and we had been studying parasites. And in that context of one of the trips to Madagascar, I made a realization that I need a microscope that has a field of view of 1 kilometer to be able to watch a single cell travel a kilometer or a particle travel this flux. If I go to my department chair, I didn't do that, but hypothetically, I would say I need 1 kilometer scale building. That would by itself be hard. And it was a little bit of an aha moment. What if the two ends of a very long tube are connected? That's your bicycle tire, and it's a new way of making a microscope. This is what we were doing in Madagascar, failing miserably. We made a 5-meter microscope first, and that's when this idea occurred. I just put this to annoy Arun a little bit. But how do we do long-distance traveling of single cells? So this is the instrument that allows us to do that. It's almost like a virtual reality arena for single cells, and the simplest context, this is essentially a sea star larva. You can see the dish that it's traveling is the size of this small bicycle tire. And if something goes up, we move the wheel down. If something goes down, we'll move the wheel up. So that's what we have done. That's what the instrument looks like. The community calls it gravity machine. I don't like the term. We're not turning off gravity anytime soon. This is the type of data sets that come out of it. You can literally watch now for the first time cellular scale interactions in the context of the ocean. And sometimes the wheels explode. But one of the things that we do is we measure all the parameters that are in the ocean, and we input them in the wheel. That's what I mean by virtual reality. So this is mirroring pressure, it's mirroring temperature, it's mirroring salinity, any sets of parameters that we care about. What have we been trying to do with it? The first thing is we went back to coccolithophore. We've been studying coccolithophore viral interaction to understand whether viral infections, which would be pandemics in the ocean, might actually be fastening carbon sequestration. This is an expedition that's ongoing. We have an expedition coming up in this summer for a month, but we now have the capacity to actually measure even single coccoliths sinking down into the ocean. The second thing that we've been doing is actually studying how alkalinity modifications work. So when you think about throwing all kinds of rocks to modulate alkalinity, you need to understand where it's going to land, because all of that is dissolving. In fact, we don't have these types of dissolution assays. So what you saw on the left is actually you can watch a single sugar cube. This is what you did probably today morning when you threw sugar in your coffee, and the rate of absorption and the rate of dissolution. But one of the most important things around all of this, and of course we measure this for all kinds of plankton. You're seeing this is what I wanted to show you about diatoms. On the right, you are watching a single diatom that within 100 milliseconds is going to pause. At this point, we have a data set of roughly around 600 species across the planet. It's the largest behavioral data set ever collected for something like this. This is a very surprising plankton. It's actually rising, except it has no cilia, no flagella. So there's this incredible biology at play. But one of the things that was important, let me just skip this, there's a lot of conservation-related work that we do, is going back to this little video. So we talked about carbon sequestration. This is actually how carbon sequestration works at a microscopic scale. You're seeing a 200 to a 500 micron particle that's essentially sinking. We can watch the sedimentation rate. And in this process, we actually discovered something very important, which we have started calling comet tails, and I'm just going to show that to you here. We have thousands of these data sets. It's the largest data set of this kind. But notice something behind it. If ever you've taken a fluid mechanics class, you should realize something is not right. The biomass, or the little flux behind this particle, is actually traveling with the particle. That implies there is something invisible that is slowing this down. And that turns out to be comet tails or mucus tails. What the bacteria are doing are generating parachutes. So you have something that will do 200 meters per day sinking, and this goes down to 0 meters per day. So the bacteria synthesize this EPS, or these mucus parachutes, and they hydrodynamically, because of the drag on this object, will completely shut down sequestration because they don't work for us. We wish they did, but they don't. And actually, it's an evolutionary advantage because most of those bacteria don't survive in deeper waters of the ocean. So it's fantastic for them to keep the food to themselves. Now, we've done this for 5 expeditions. We're mapping up all the data. And I'm going to shut down very soon. But I just want to show you this. What does the estimates look like? What this does is it shows that the residence time of marine snow doubles. But just like Noah brought up an exponential, there is an exponential in this, because if this mass stays in the water column for long, it gets eaten by bacteria and it gets remineralized. Nothing gets fixed. So you have something where a high amount of sequestration is happening all the way to no sequestration happening. We did these budgets, and the current estimated budget error for this is essentially astronomical. When we do these errors, this is roughly equivalent to 40 ppm of CO2 as an error bar. The last estimate that was done in that, people had been thinking about, okay, maybe we have a 10 ppm. I mean, that's 80 gigaton of an error in how we are thinking about the carbon sequestration works. There's lots of challenges in terms of thinking about where the hell is the carbon going then. Maybe there are other sources that we don't know about. But again, this is the value of building bottom-up in terms of data. And I think I just want to end with this one last image, because I know I'm out of time. One positive note. The positive note is actually very recently we've also discovered a plankton species that now increases sequestration. These are radiolaurin, so we are heavily working on this now. This happens to be in our backyard in California. And so it's not all doom and gloom. You just have to go and see what happens out there. And then the last thing I want to mention, going back to this tool and democratization, one of the most important things for us in terms of measurements and for any kind of model that I care about, we need to massively increase our capacity to measure on this planet. Many of you have been talking about this. We released this open source tool less than a year and a half ago. Just plans on a website. It takes a day or two to put together, and what we started seeing, this is the type of data sets that you just saw. much of this data has incredible bioindicators, but something really interesting happened on the community side. I want to go back to what I said about full scope, is this happened. I started getting messages from people, their garages, many of these people are not scientists, but they deeply care about their world and they want to contribute. And because the machines are autonomous, they actually collect quantitative data that can go in the model. And this has been growing and growing and growing. At this point, there are 300 of these units out there. We're starting to build more of them, and 25 million data sets have now been actually put in the ecotaxa just from this community alone, where effectively all we started doing was really build open source tools around measurements. So this is something that I'm very excited about. And one thing I want to end with is we have 2 of them floating in the Bay Area that are completely autonomous now, and we are launching a new entity. We launched Planktoscope some time ago, but we're launching what we are calling Pelagica, which is now a completely autonomous. This couples to the SOFAR buoys. That's another low-cost instrumentation company that's out there that gives us power and communication. We had edge computing on this as a platform. And for hundreds of applications in terms of mapping kelp farms, all the way to mapping bio reefs, for calibration of satellites, something that we essentially can do in a completely autonomous way. I'm out of time. I'm going to close by just saying something on kind of sets of discussions about our capacity. It does cost a lot of money to be at sea, $60,000 a day. That's why we do need autonomous platforms. That's why we need frugal instruments. But our capacity to observe our planet is actually also shrinking. If you look back in 30 years ago, we had more naval vessels, much more capacity to be at sea. And so it's ironic in some sense. And at that same time, the dialogue between basic science, we are trying to understand something that we are also trying to fix. And I think this is really one of my messages. I am all for all kinds of technologies because as Noah's carrying me, we have no choice. But at the other hand, we have to build them with basic science as a context, measurement as a context. And it's not something that every time you go out measure, not only do you learn whether you're doing the right thing, but you actually also get completely new ideas that you would not get otherwise. Sorry for running out of time. I'm around if people want to play with the tools. You can come and play.